Two small cups are positioned side by side on a preparation table. A bag of soil is opened, ready for use. Soil is carefully poured from the bag into the first cup. The first cup is filled nearly to the top with soil. The process is repeated, filling the second cup with soil as well. The process begins with the selection of suitable seeds, such as beans or peas. Prepare two separate cups, each filled with appropriate soil. Take one seed and gently place it into the soil of the first cup. Ensure the seed is properly positioned for optimal growth. Repeat the process by placing the second seed into the soil of the second cup. This careful placement is essential for successful seed germination and plant development. The initial watering is a crucial step in starting the growing process. Begin by filling a watering can with water. Ensure the water is poured evenly over the soil in the first cup. The goal is to thoroughly moisten the soil, not to flood it. Observe the soil to ensure it is absorbing the water adequately. Note that the second cup remains dry as a control or for later use. The experiment requires both cups to be placed in a sunny location. A windowsill is selected as the location to maximize sun exposure. The cups are positioned side by side to ensure they receive approximately equal sunlight. Equal sunlight exposure is crucial for a fair comparison in the experiment. Two cups containing soil samples are being compared. The soil in the first cup appears moist. The moist soil exhibits a dark coloration. In contrast, the soil in the second cup is dry. The dry soil has a light coloration. This visual comparison highlights the difference in moisture content between the two soil samples. The experiment involves observing the effects of different watering routines on two separate cups. Cup 1 receives a second watering, following the established routine. Cup 2 remains untouched, serving as a control for comparison. Time-lapse recording captures the subtle changes occurring in both cups over time. Observations are being made on the first cup of seeds. The soil in the cup remains consistently moist. Subtle changes are now visible in the seeds. Slight swelling of the seed can be observed. These changes indicate the initial stages of germination. A second cup is being observed as part of an experiment. This cup is maintained in a dry condition. After 4 days, no visible changes are observed in the dry cup. The soil remains completely dry to the touch. The seed within the dry cup shows no signs of germination or alteration. The emergence of a sprout from the soil signifies the start of plant growth. This initial stage is critical for the plant's development. The sprout indicates successful germination. Further growth will depend on environmental factors. The initial watering involves saturating the soil in the first cup. This ensures the soil reaches an adequate moisture level for initial hydration. Subsequent watering maintains the soil's moisture, preventing it from drying out. Consistent moisture is crucial for successful plant growth and development. The observation takes place on day 7 of the experiment. The second cup, intended for growth, remains barren. There are no visible signs of plant germination or sprouting. The soil in the cup appears visibly dry. Cracks are evident on the surface of the soil, indicating a lack of moisture. The sprout exhibits continued growth in height. This indicates successful germination and nutrient uptake. Small leaves begin to emerge from the sprout. Leaf development signifies the start of photosynthesis. The sprout is transitioning from seedling to a young plant. A side-by-side comparison highlights the variance in seedling development. The first cup demonstrates successful germination with a visible seedling. The seedling in the first cup shows signs of healthy initial growth. In contrast, the second cup shows no signs of germination or seedling emergence. The absence of growth in the second cup indicates a potential issue. This visual disparity underscores the different outcomes of the germination process. The seedling's leaves are undergoing expansion. This expansion indicates healthy growth. The leaves exhibit a vibrant green color. The color suggests adequate chlorophyll production. The seedling is progressing well in its development. The experiment involves observing soil conditions in 2 separate cups. The soil in the first cup maintains a consistently moist state. In contrast, the soil in the second cup is completely dry. This contrast highlights the difference in moisture levels between the 2 soil samples. A seed in the dry cup exhibits signs of withering. The seed appears shriveled. The seed also appears dry. These observations indicate a lack of moisture. The lack of moisture prevents proper seed development. The seedling exhibits continued growth. More leaves are developing on the seedling. This indicates healthy plant development. The seedling is thriving in its current environment. Continued observation will track further progress. The seedling has developed into a small plant. Consistent watering supports the plant's growth. The plant is thriving in its current environment. This stage represents the final growth phase. The plant is now more robust and developed. Continued care is essential for optimal development. The sequence visualizes the complete growth cycle of a plant. The process begins with a seed. The seed germinates and sprouts. The plant develops its initial leaves. It continues to grow and mature. The final stage shows the plant as a young seedling. A side-by-side comparison highlights the critical role of water in plant development. One cup contains a plant that has received adequate hydration. The hydrated plant exhibits healthy growth and vibrant foliage. The other cup, lacking water, contains no plant. This stark contrast visually demonstrates water's necessity for plant survival. Water is essential for nutrient transport, photosynthesis, and cell structure in plants. The root system is a vital component of a growing plant. It is primarily responsible for absorbing water from the surrounding soil. This water absorption is crucial for plant hydration and nutrient transport. The animation visually demonstrates the root system's water uptake process. Water molecules are absorbed from the soil by the plant's roots. Water then moves through the roots into the plant stem. From the stem, water is transported upwards to the leaves. This process demonstrates the pathway of water transport within a plant. Photosynthesis is the process by which plants convert light energy into chemical energy. Water is a crucial reactant in the photosynthetic process. Plants absorb water through their roots. Water molecules are split during the light-dependent reactions of photosynthesis. This splitting releases electrons that are essential for the production of ATP and NADPH, which power the creation of glucose. Water is essential for maintaining the turgor pressure within plant cells. Turgor pressure refers to the force exerted by water against the cell wall. This pressure helps keep the plant cell firm and rigid. Adequate hydration ensures that plant cells remain turgid. Turgidity provides structural support to the entire plant. Without sufficient water, cells lose turgor, leading to wilting and reduced structural integrity. Water is an essential requirement for plant growth and survival. Seeds require water to initiate the germination process. Germination is the process by which a seed develops into a seedling. Without sufficient water, seeds will fail to germinate. Established plants also depend on water for various physiological functions and overall survival.